Spot the difference between these two bananas. While it's healthy for a banana to have some spots while yellow and ripe, deep brown spots on a green banana are a symptom of the disease anthracnose. Anthracnose is a class of fungal infection commonly caused by the fungal pathogen Choledotrichum musae. It often affects bananas post-harvest and can be seen through those sunken spots on the green flesh. But how does anthracnose speed up this decaying process so much faster than a banana can do on its own? It's time to get technical for a second. Let's start by zooming in on our banana a little bit. And right there. Stop. This is one of millions of cells that comprise the banana. And here's what's going on inside that cell. This is a cell communication pathway. It is made up of the steps of how cells send signals to each other. All signaling pathways start with a small molecule called a ligand. The ligand binds to the surface of the cell. This activates something called the G protein. Once activated, it will send another molecule to activate phospholipase C, or PLC. Remember this molecule. It will be important later. Now, we're not going to explain the entire pathway because there are literally hundreds of different things that can happen after this. But it is important to know that once it is turned on, PLC can go on to activate secondary messengers, such as those seen highlighted here. At the end of the day, the big takeaway is that this pathway ends by producing some kind of cellular response, which helps keep the cells of the banana functioning properly. Researchers in China found a few years ago that when there was increased anthracnose infection, there was increased expression of PLC. They theorized that somehow the function of the PLC enzyme was linked to the spread of anthracnose. But they weren't able to answer one question. How? Was PLC working against the anthracnose, or was it actually helping it to spread?
That's where our team comes in. In part 1 of our project, we're going to be using gene knockout. Banana DNA has genes that code for the molecules in the PLC pathway, including PLC. We want to see what happens when we knock down either single genes or combinations of genes involved in the pathway and add anthracnose. Will anthracnose still spread if those molecules aren't available for a brief period of time after infection? Note the difference between gene knockdown versus knockout. Knocking down temporarily stops production of a gene product, while knocking out permanently deletes the gene for good. The banana needs the phospholipase pathway for many different signals, so it's important that we don't eliminate these genes entirely through knockout. That leads us into part 2 of our project. All enzymes have active sites that they use to bind substrates. If we either delete or replace amino acids involved in the binding of PLC to its substrate, what will happen? We want to know, will anthracnose spread less efficiently if PLC is mutated and loses its catalytic efficiency? Now for part 3. In part 3, we're going to take a slightly different approach. Some species already have resistance to anthracnose infection, like the common bean. The common bean actually has genes, no, not those kind of genes, that are coded with resistance. We want to see if it's possible to knock in or add resistance genes from the bean DNA into the banana DNA. To do this, we're going to construct something called a gene pyramid. Some of these known resistance genes don't work without each other, and some work together better than they did separately. In order to create the best genotype, we're going to see how random pairings of resistance genes work when investigated together. By experimenting with different pairs of genes, we can determine the gene combination which has the most resistance.
At this point, you might be wondering how exactly anthracnose plays a role on a global scale. Bananas are the 4th most consumed staple crop after rice, wheat, and maize. They're also a crop that many developing countries currently depend on to support their economies. About 20 to 25% of the world's harvested bananas are decayed by different fungi during post-harvest handling, and it just so happens that anthracnose is one of the leading diseases associated with fungal infection among bananas. This means that banana growers are in need of solutions to improve crop yield and prevent waste. Right now, Millions of banana farmers are depending on research like ours to maintain their crop survival rate and uphold the major state of their economy. Our project ultimately aims to better tackle that problem, one gene at a time.